have you started looking at it at all?
Yes,
Yes.
ours is kind of, I don't know, maybe I'm doing this all wrong, but when I got to the final graph, we have like our 80% is like at way, way up high, and then our 100% is like
Right.
halfway.
Right. So what do you think is going on there?
Well, we probably had an oxygen leak.
Yeah, I suspect there was an oxygen leak,
Yeah,
or
similar thing. Yeah, we had the same. I had the same in mind.
right? Or I mean, it can also be, human error, people messing up while they're mixing things up, or it could be some of the solutions were like I tried to refill a lot of the hydrogen peroxide with new stuff, but I don't trust some of those old ones weren't there from a year ago. Because some of them, when I was realizing they were not all freshly filled, some of them were just kind of filled. So there's this is at sources of variability. Like in your discussion, you most definitely need to address this kind of thing. If you get results that really are not what you think you should find, then you still need to report what you found, but you do need to bring up in your discussion, like how that's different from what you expected and what you think might have happened. That's part of a good discussion, is owning what's real. You poke at the physical world and it wiggles in some way and you have to describe it, but that doesn't mean that you have to just take it at face value. You can like talk about these sources of variability and things that might have gone wrong. What other things did people notice?
I found figuring out the best of line to be very, like, I was really unsure as I was doing it.
Yeah, the best fit line can be really tricky. Sometimes even like, two different people who are being kind of responsible and skilled will come up with slightly different lines. Part of the problem with this lab is the bubbles are so imprecise, like a big boop, and so the true value of the graph might be like 5 mils above or below the actual dot because the bubble was just about to come up or hasn't come up yet, or so there's that part which makes this tricky. You can usually get pretty close. What other things did people notice?
Just when plotting out the oxygen evolved graphs, in some of the higher concentration solutions, it'd be going and going, going, and then level off, and then get going again, just toward the top, maybe the last third of it. So I was thinking that one possibility is that maybe the person stirring it was slacking a little toward the end. No, I'm not trying to name names, but I mean, would there be?
Thank you.
I would say.
Sorry to throw you under the bus.
It's very likely that some unnamed person was kind of, you get also, it's exciting to like, is this person going to actually read off the bubble or are they going to spaz? And, and you're watching them and you get excited and then you kind of forget. It's like, oh, wait, I'm supposed to keep this going. And then as soon as you start agitating again, the reaction rate will probably increase. So I could easily see that happening. Or else maybe they get excited and squeeze and push there was a little leak and now it's not a leak anymore. Who knows?
So I still have not actually gotten the best fit lines, but in our group, it seemed like to the extent that was happening, like I said, it was pretty close to the end anyway. Didn't turn out to be quite the end, but there's probably still enough data to get a good line, just looking at the first, the middle part.
So that is definitely true. So thing to remember when you're doing these best fit lines. The reaction is going to be going at its best in the beginning, probably the first 30 seconds. And it will slowly start, it doesn't go like this. Oops. It doesn't go like up and then die. It goes up and slowly dies and slowly dies and slowly dies. It's more like that. So when we see one of these things, it's kind of like that. And what this means is my best fit line is probably around here. This place where it's starting to lose steam, I don't care about that anymore. This is usually like from 35, 40 seconds on. It's, even if it's still going, it's in the process of slowing down. So that is not the place where I want to consider for my best fit line. I do not want to draw my best fit line like this or something. It's going to be when the thing is really going. So like John, to your point, if it kind of does some weird stuff up here, it's irrelevant. It's not going to affect your analysis at all. So what else did people notice when they're starting to analyze this stuff?
I got the same slope for trial 4 and trial 5, and I'm wondering if it's because I did a bad or inaccurate best fit line for those two, or if maybe there's a reason why I got, I'm wondering if my group also got the same slope for trial 4 and trial 5, or if I need to redraw the line.
So did anybody
I
else?
got totally different one.
You
I
did?
got, like I was saying, I got like one that was like completely different than the other for the last two.
And we have
And
the
then
same
I got the
data.
same in 3 and 4, and we were in the same group.
Yeah. Oh,
But
no,
then I
didn't-
got the 5th one went down about 20% for me.
Yeah, that was what I'm talking
And
about.
and then I checked the slopes at different points on my line, but I got the same thing.
What?
So all three of us are in the same group. So I'm just wondering how we can be so off.
It doesn't sound like you're that far off, actually. Yeah, in fact, let's. So what did it look like from like 20 to 40%?
I had it go from 0.4 to 0.5.
It went up a bit. How about you, Catlin?
I got for 20%, I got 0.25, and for 40%, I got 0.45.
You have a bit more of it. So obviously you guys have drawn different best fit lines. It'd be interesting to actually see what it's kind of hard to do.
Similar to mine too. Those are both similar. I'm in the same group.
So and is that going to be consistent with your initial hypothesis?
Yes.
Yes.
Yeah, we're adding higher concentration of substrate. We're seeing a faster reaction rate. What Catlin is saying is up here, these stayed the same. And actually, I think it was Jen was saying these stayed the same, and this even seemed to go down a bit.
Yeah, I was expecting the last two to stay the same. So I was kind of surprised then when the last, because I thought maybe it had reached like saturation or something.
I mean, what probably happened if these are not increasing anymore? What's probably happened? You said
Saturation.
Saturation, yeah.
The evidence that we've hit saturation is that it's not getting faster. If something goes down a bit like that, I would say it's probably just the noise. I trust, I would say any of these things that you calculate is going to be, some range of what is it really, based on just the wiggle room and where exactly you draw your best fit line, the wiggle room in those, just how sloppy the bubbles are and everything. So I would say if you have an actual dot on your thing, the actual dot is probably, kind of about where the real number is. The real number is probably in some error bars around that dot. So if you went down, I would say that doesn't make me think that something is horribly wrong there. It's just that it's probably within the margin of error, even that this is still kind of making me think, like, supporting that we're at some kind of saturation if it's not speeding up and they're relatively close to each other up there, rather than kind of increasing as we increase the concentration of the substrate.
The two of mine that were actually the same were the 40 and 60, and then it jumped up at 80. But I still think that supports, pretty much still supports what I was thinking.
So, yeah, and I would say there's also weirdness again in the fact that people got their 100% had less, or made less by the end than the other. That's weird. So there's other sources of variability that also make us a little doubtful of how much we can rely on any particular of these dots. Because ideally, theoretically, if I'm looking at time and seconds and O2, mils of O2, it's going to go up, it'll kind of die. This might be the 20%, trial 1. If I have trial 2, what would I expect the graph to look like theoretically?
A stronger slope.
Be a steeper slope, go a little longer, and by the time it ended, we would have made more oxygen. We had twice as much H2O2 to start with, so we're gonna have twice as much oxygen by the time we're done with breaking down it and making oxygen bubbles. So this would be like trial 2. Trial 3 is gonna probably be steeper or even, and make even more bubbles by the end. If we're at saturation, what would trials 4 and 5 look like?
The same.
Same what?
They'd have the same rate or
Yeah.
same slope.
Be the same slope. The slope would be the same, but what would be different?
It would make more oxygen since there was more reactant.
Exactly. Since there's more oxygen. Well, yeah, so there's same rate, but by the time you're done, we'll have made more oxygen. So same slope would mean we're at saturation. The rate is the same, but you'll still end up making more oxygen by the end because you have more hydrogen peroxide to start with. So more oxygen is going to be made by the time it's all broken down. So this is theoretically what we'd find. When I looked at, in fact, I can share now. I'll share, except for whoever didn't send in the thing. I've got 4 of them. I have here for trial 1, trial 2, trial 3, trial 4, trial 5. Basically what I did was I put in all of your data. I got the average of that data. And then oxygen involved is just 50 minus whatever that burette is. Again, if we look at the data, this group probably should have thought twice. Like if you have nothing changed, probably something was wrong, right? So their group 1 data just went and never ended. Like if you're doing an experiment and the result looks pretty weird, you probably want to do that trial again. I put it in here just for completeness, but I would probably, in fact, I might just kind of get it out of there right now because it's pretty clear that trial was not a valid trial. But here, this one, we can see most people for trial 1 ended up with about 30 mils of oxygen by the end. Trial 2, still about 30 mils. That's kind of weird. Trial 3, I'm sorry, I take it back. Mils of oxygen. We ended up with 17 mils of oxygen by the end of trial 1. 20 mils of oxygen, better. We've got more oxygen for trial 2, even more oxygen for trial 3, even more oxygen for trial 4. Trial 5 is the one a lot of people ended up getting less. I'm not sure what's going on there. And then trial 6, everybody got that kind of classic thing. You know, if we look at it in terms of, this is just the average of everybody's stuff, we actually see it, actually, except for the total freaking weirdness about trials 4 and 5, right? You know, trial 5 should be at the top, trial 4 should be here. But aside from that, we actually do see something that looks relatively compelling, where, you know, if I kind of look at this and I kind of look at this, that's getting a little steeper. And then these are, you know, looking a little more like they're going to, you know, so I have to say, in just general, I do see something that looks like saturation if I'm just eyeballing it. This one, on the other hand, what do we see for the one that was pure distilled water? What's the slope on that one?
0.
It's 0, right? Because remember, we're going to throw out the weirdness in the beginning because we know that there's that 5 mil bubble. We talked about that last class already. You know, one thing I did just as a, yeah, as a way to look at it as well, this graph here, what I did was I just looked at the data from 10 seconds to 35 seconds. This is not as good as doing it by hand, but I just did this as a way to quickly do this on the screen. You know, and this is actually showing, it calculates the slope. If you show the formula of the best fit line, and remember y equals mx plus b, it's just your arithmetic days. So here it's showing that the slope at trial 1 was like, you know, 0.37, but it was 0.5. And then it's all around a 0.6 more or less for the upper 3. So there's, you know, there's something relatively compelling. Again, the fact that trial 4 and 5 are reversed in terms of how much oxygen's being made is weird. It's making me like a little uncomfortable.
I have a question. There's nothing like the osmotic pressure that we saw in the osmometer lab that has to do with the oxygen weighing down.
No, nothing like that.
I was wondering, could it be that the enzymes in the pH solution have settled to the bottom, and then when you're collecting them, you're not getting as many enzymes towards the end?
No, it's a solution. It's not going to be a suspension. So I don't think that would be an issue. You know, it could be that the flask wasn't properly cleaned, or there was some residual stuff in the flask, for instance. That could cause problems, contamination of the flask that you're mixing things up in. So there's so many things that can go wrong. I would say if I got this data and I really cared and I had more time and this was more my actual research, I would say, okay, this is interesting, but I need to do this again before I really trust what's going on here. You know, and also I would probably redo the bubbles. Like, again, we didn't do it this time, but Tina and I have been experimenting running the bubbles through a little piece of cloth so they end up a bit finer instead of those big gloopy bubbles that go up the tube. because then you don't have those big discontinuities as like a big 5 mil bubble comes up all at once or something. So just kind of, so I did want to kind of show you though the basic trends once we put like everybody together, and I will put this up on the module. I don't have it there yet, but I'll put this data there just so you have, I still would like you to draw your best fit lines and do your work for your data. But then you can have this as something to compare to. But this is not instead of, I know some, some of you, I appreciate that some of you guys have been on it and have done your initial analysis. If you have not done it, please do it. And if you're running into trouble and getting confused, that's more reason to do it now and know you're getting confused and you can reach out and get some guidance. Like I mentioned last week, this is one where people often get confused. That's normal. But if you are confused, you need to, find resources, including office hours and things like that, to make sure that you understand how to analyze the data. The grading of this lab, you'll get like a couple of, a point or two for having an objective and getting the data. But the real meat and potatoes of your grade for this lab is, did you understand what you're doing? Did you analyze the data and interpret it and see what it means? So don't think just by having like a bunch of charts full of numbers that you're pretty much done. The real journey with this lab begins after you got all of those numbers and you start interpreting them. So just kind of putting that out there.
Do we need to include this class data in a write-up? Or
yeah, in general, it's a, or just like the graph, for instance, and it could be either one, just as a way to give some context, like especially like for the group who had basically a flat line for trial 1. This is going to be an important thing for you all to realize that, oh, something was really wrong. That's not what everybody else got. Like, so in your discussion, you're going to have to say, like, even though our trial 1 showed like no reaction, that wouldn't be expected. And in fact, none of the other groups got that as well. So something must have gone wrong. And like I said, you don't lose points for having something gone wrong. You lose points for not bringing up in your discussion that something's gone wrong and what do you think happened, right? Yeah, that's the thing I think important in these experiments is the important part is the discussion. Like, what does it mean? So any questions before we move on from this one? Yeah, if you have not analyzed it or you're getting intimidated by it or whatever, please don't put it off too long, because if you wait till the very end, it'll be too late. Okay.